A CSUN biology team says it has discovered a new cancer research method.
Their research was published in the journal OncoTarget. Here's Morgan Bertolotti with more.
Welcome to Studio B. This is Morgan Bertolotti. I'm sitting with CSUN biology professor Jonathan Kelber. How are you today, Jonathan?
I'm doing good. Thanks for having me.
Excellent. Thanks for coming. Jonathan, along with 2 graduate students, have developed a novel method for extracting genetic data out of archived pancreatic cancer tissue. So extracting this information out of these cancer samples has been a major bottleneck in oncology research until now. You use the microhomogenizer. How does this effectively free the trapped molecular information?
When a cancer patient goes into the doctor and they effectively remove the tumor, that tissue is sent to the pathology lab, and the pathologist fixes the tissue, and then they embed it in a formalin-fixed paraffin block. That's great for the pathologist, but for future studies on that tissue, it makes it very difficult. And so what we've discovered is that our collaborators at Claremont Biosolutions, they have produced this really great tool. It's called a microhomogenizer. And the tool disrupts the tissue, and it disrupts the paraffin and breaks it apart. And we believe that what's happening then is it's freeing or liberating more of the molecular information.
What are some of the challenges that you face when using the formalin-fixed paraffin embedded cubes?
When you formalin fix anything, the goal is to kill it and to fix it, as the name implies. And what that does is it cross-links a lot of the biomolecules. When those cross-linking steps occur, it prevents downstream applications which require the liberation of these biomolecules. But they've been really inefficient. So our method, I think, improves quite significantly the efficiency with which we are able to get out these important biomolecules for downstream study.
Well, I understand there are 2 different types of environments. There's a subcutaneous environment versus the orthotopic. Can you kind of explain the differences and the challenges in there?
We want to study the cancer cells. We want to evaluate what genes are contributing to the cancer. And one of the ways we do this is by putting human cancer cells into animal models and studying how the cancer cells grow, how they divide, how they spread or metastasize, as we call it. We decided to apply this new FFPE RNA extraction protocol using microhomogenizer to discern the differences between gene expression or genes being turned off or on when the cancer cells are in the orthotopic microenvironment versus the subcutaneous. And we saw very, very striking differences. It really supports the idea that we need to be studying these cancer cells under the most physiological systems possible.
And you mentioned that you've been working on this for about 4 years. Where do you go from here?
We know that there are tens of millions of these FFPE samples around the world. And so the more samples we can access and the more data we can produce on a large scale from these samples, we'll get a better picture of what otherwise is a really devastating disease, pancreatic cancer.
So when might some patients see benefits from this?
Timing-wise, I think, it could be on the diagnostic side, it could be as early as, a year to 5 years. On the kind of prognostic side, where you're predicting based on gene expression and how to treat patients based on their specific disease, I'm thinking maybe more 10 to 20 years. It takes time for these types of advances to progress through the proper channels and to be available for clinical use.
Thank you for joining us here today. This has been Jonathan Kelber, CSUN biology professor, and this is Morgan Bertolotti in Studio B. Back to you in the studio.